A sunlit suburban garden appears serene, almost mundane. A robin hops across fresh mulch. A blue jay flashes across the tree line. A house sparrow plucks a dandelion seed. But beneath this surface of tranquil routine lies a quiet, calculated war against microscopic invaders and a sophisticated biological intelligence at work. For centuries, orthodox biology viewed animals as passive recipients of disease, relying entirely on internal immune systems to battle parasites, bacteria, and toxins. But field ethologists and neurobiologists tracking wild populations have uncovered a far more staggering reality. Birds do not merely endure illness, they actively diagnose, seek out, and administer external chemical compounds with surgical precision. Consider the European starling selecting nesting material in early spring. The bird bypasses dozens of lush green leaves to deliberately harvest the aromatic sprigs of wild carrot, yarrow, and lavender. This is not decorative instinct. Laboratory analysis reveals these specific flora contain heavy concentrations of volatile secondary metabolites, essential oils that inhibit the hatching of blood-sucking mite eggs and suppress bacterial growth. The starling is not just building a cradle, it is fumigating a nursery, calculating dosage and chemical potency long before its offspring hatch. This phenomenon, known as zoopharmacognosy, challenges the foundational assumptions of animal cognition. Self-medication requires an organism to recognize a physiological threat, locate a precise chemical agent in a vast ecosystem, and apply it in a controlled manner. It demands an evolutionary memory that functions as a living, outdoor pharmacy. In the humid canopies of South America, red and green macaws execute a daily routine that baffled early naturalists. High above the forest floor, these massive parrots consume toxic seeds packed with lethal alkaloids, compounds designed by plants specifically to destroy an herbivore's liver. Yet the macaws suffer no ill effects. Following their morning feed, entire flocks migrate to exposed riverbanks of bare, mineral-rich clay. They scrape away the surface layer and ingest precise quantities of sodium-rich bentonite. The clay acts as a chemical sponge, binding to the alkaloids within the bird's digestive tract and neutralizing the poison before it enters the bloodstream. The bird balances poison with antidote, navigating a lethal dietary tightrope with absolute accuracy. The sophistication deepens when we observe urban adapters. In dense metropolitan centers, house sparrows and finches have been documented harvesting discarded cigarette butts to weave into their nest linings. What initially seemed like a hazardous ingestion of human trash revealed a stark chemical strategy. Nicotine is a powerful botanical insecticide. Nests woven with nicotine-laden filter fibers show a drastic reduction in parasitic tick infestations, directly increasing chick survival rates. The birds have co-opted synthetic industrial waste, recognizing its biochemical utility and integrating human pollution into their medicinal repertoire. Even more enigmatic is the widespread, deliberate behavior known as anting. A songbird alights on an active mound of wood ants, spreads its wings flat against the earth, and allows hundreds of agitated insects to swarm its plumage. Alternatively, the bird will grasp a single ant in its beak and methodically rub the insect along the shafts of its flight feathers. As the ants defend themselves, they spray concentrated formic acid. This corrosive liquid dissolves feather parasites, neutralizes harmful fungi, and acts as a topical antiseptic across the bird's skin. The bird endures the brief sting of chemical defense to secure a long-term hygienic advantage. These actions are not random trial and error. They represent a complex interaction between genetic programming, environmental observation, and sensory feedback. The avian brain, far from being simple or primitive, contains hyper-specialized neural circuits capable of processing complex biochemical cues from the environment. A bird evaluates the scent, taste, and tactile feedback of a plant or insect, cross-referencing sensory inputs with its internal state. The implications ripple across neurobiology and evolutionary theory. If a bird can select specific botanical compounds to alter its internal biology or protect its environment, we are witnessing an active, cognitive intervention in the process of survival. The wild is not merely a stage where animals search for food, it is a vast, dynamic apothecary, and birds are among its most adept physicians. Beyond the immediate physiological relief lies a far more profound cognitive mechanism, the capacity for cross-generational transmission and cultural learning. The acquisition of pharmacognostic knowledge is not merely an automated genetic legacy hardwired into avian DNA. It is an active, socially learned tradition transmitted through observational learning across flock hierarchies. When a dominant female European starling selects specific sprigs of wild carrot or elderberry foliage to line her nest, juvenile birds within the colony observe and record the specific visual markers, tactile textures, and volatile olfactory signatures of those plants. Subsequent generations reproduce these precise foraging choices with meticulous fidelity, establishing localized botanical traditions unique to specific regional populations. In regions where urbanization or climate shifts alter local flora, corvids and songbirds have been observed rapidly updating their medicinal catalogs, testing novel plant species and integrating them into their nesting routines. This dynamic adaptability highlights an intricate sensory architecture. The avian olfactory system, long dismissed by 19th century anatomical paradigms as vestigial or rudimentary, is in fact acutely tuned to complex biochemical signatures. A blue tit or starling flying through a dense forest canopy does not perceive a uniform wall of green foliage. Instead, its olfactory receptors process a multi-layered chemical landscape, detecting subtle concentrations of monoterpenes, phenols, and alkaloids drifting on the morning air currents. The bird parses this invisible fragrance map, isolating a single patch of aromatic yarrow amidst hundreds of non-medicinal plant species with absolute precision. The sophisticated nature of avian self-medication extends beyond preventative nest hygiene to targeted internal treatment. When parasitic infections burden an individual bird, its internal physiology triggers shifts in appetite and taste thresholds. Ethologists studying wild songbirds have documented instances where infected individuals actively seek out bitter, secondary metabolite-rich berries and roots that healthy members of the flock explicitly avoid. The bird willingly consumes low doses of plant toxins, compounds that would cause physiological stress in a healthy organism, in order to purge intestinal helminthes and protozoa. This deliberate alteration of dietary preference in response to an internal infection represents an extraordinary level of homeostatic regulation, guided by subtle neurochemical feedback loops. Consider the intricate chemical engineering exhibited by wood pigeons during seasonal transitions. In late autumn, as temperature shifts increase the prevalence of systemic fungal spores in damp leaf litter, flocks congregate around wild oak stands to ingest specific quantities of tannic acid-heavy acorns. The high concentration of astringent tannins binds to dietary proteins in the avian gut, forming a protective mucus barrier that prevents fungal pathogens from colonizing the intestinal lining. The birds adjust their intake based on environmental dampness, balancing the metabolic cost of processing bitter tannins against the immediate threat of lethal fungal invasion. Even the phenomenon of bathing takes on an entirely new dimension when viewed through the lens of ethological pharmacology. A house sparrow plunging into a pool of stagnant water, followed immediately by a vigorous dust bath in fine mineral-rich soil, is not simply removing surface dirt. The fine clay particles absorb excess prion gland lipids, while specific soil minerals act as natural desiccant agents, dehydrating the soft exoskeletons of feather mites and lice. When birds follow this with sunbathing, spreading their flight feathers wide under direct ultraviolet light, the heat triggers preen oil chemical conversions, transforming preen gland secretions into active vitamin D compounds that are subsequently ingested during preening. The broader scientific community is only beginning to decode the vast biochemical library utilized by wild birds. Field experiments combining high-resolution GPS tracking, mass spectrometry, and video surveillance continue to reveal new dimensions of avian zoopharmacognosy. From corvids utilizing pine resin as an antimicrobial adhesive in cold climates, to tropical hummingbirds selecting nectar sources rich in natural immunostimulants, the evidence paints an unmistakable picture. The perception of birds as simple, instinct-driven creatures governed solely by immediate survival reflexes is fundamentally obsolete. What we observe in every suburban park, Dense forest and urban roofscape is a continuous, highly refined practice of natural medicine. Birds navigate their environments as active chemical strategists, analyzing, experimenting, and applying the biochemical potential of the natural world to survive against relentless biological pressure. The skies are populated not merely by instinctual foragers, but by the ancient, silent apothecaries of the natural world,